Nice. Okay. I think this is here.
Okay, so I'm actually, apparently it's not typical in physics, but I'm going to introduce the speaker, who's been in my lab, kind of in my lab. So let me explain. I'm David Baum. I'm in botany, and that's strange. So if you don't know, I mean, Praful's kind of, I feel like it's like a melded family. It's like he has 3 different advisors, but I don't know which of us are actually advisors, but are in physics. And then I think, where's the camera? There?
Oh,
whoever it is.
yeah.
Eric Smith is a physicist who works on Origin of Life, who also has been playing a very important role, for which I'm very grateful in mentoring Praful. And so, you know, he's then also working kind of informally in my group in the WID on Origin of Life. And I guess in 2019, you were in this room, perhaps, when you talking to one of my students and heard about our interest in Origin of Life.
That's exactly how.
And he came over and was sort of like, can I come? I'm sure I can be useful. Can I come? I'm like, I don't know, you have to be useful, but you're fun to talk to, so why not? And so after whatever it is, 3 years, 4 years,
maybe
of having the pleasure of having Praful kind of in my group, I've learned way more for sure than he's possibly learned from me. But it's really kind of exciting for me to see him pull it all together into a lovely thesis and get an opportunity to present it to his own department in physics. So anyway, I welcome you all, and I'm very excited to hear Praful do this and move on. I should say he's going to stay on afterwards in my lab, which is even better.
Cool. Right. Thank you everyone for coming. So yeah, just a few things before we start. There are refreshments in the back, but I think everyone's happy about that. Yeah, feel free to ask questions anytime. That's fine. So today, my title my thesis, Mathematics of Evolving Ecologies in Chemical Reaction Networks and the Origins of Biochemical Life. So sorry, doesn't.
You need to click once on the screen, maybe.
Okay. So let's see.
No.
Yeah. So that's right. So here's the outline. I'll first tell you about my thesis question. Then I will explain what are chemical reaction networks. I'll show you this thing that we have defined as cluster chemical reaction networks. And then I will explain the notion of ecology, again, give some background on stochastic chemical kinetics, and explain our notion of evolvability. And then finally, I'll discuss a few things about the biochemical origins of life. So what is the motivating question for this for my defense and my thesis? So for the field itself, the question is, was life inevitable? So from a physics perspective, you can think of it like this. Modern physics, cosmology gives a very good picture of what happened all the way from the Big Bang to planet formation. And I think there's a light pointed over here. Okay, to planet formation.
You can use that big stick.
Oh, thank you so much.
It's also useful as a weapon.
Yes, no questions. Okay, so we know how to get from the Big Bang to the planet formation, right, in principle. And then that was around 4.5 billion years ago. And then from 3.7 billion years ago, we have fossil evidence of this earliest life form. And actually, this group, a part of it is at UW-Madison. And the idea is that, that this life form also had some notion of DNA and inheritance and vertical gene transfer. So the question then becomes, what are the origins of biochemical life, right? How did Earth transform from an otherwise inhabitable planet to one with life? And so this is the question of the field. That's what the field of origin of life is about. My question is something which is much more smaller. The thing that I want to understand is this: okay, all life as we know it has this it has DNA. It has these long polymers through which information is encoded, and then the next offspring comes into being, and so on. So the question is, though, in an environment with only small molecules, can we have long polymers emerge spontaneously? And even if they do emerge spontaneously, can we have big enough, high enough abundance of these long polymers such that in a stable way? And I will of course explain these things. And putting and saying all of this in the language of the thesis itself, could an ecology of polymers spontaneously emerge and directionally evolve towards higher mean lengths, right? So all I'm asking is just a question about whether these long polymers could have come about and sort of stably maintain their concentration. So, this is just a broad overview. Before we get into the mathematical details, I first want to say a few things about life itself. So defining life is also, of course, a very it is very tricky. But what I will try to do over here for the scope of this presentation is we will define a concept of life through only necessary conditions. And since we're restricting our discussion to the origins of life, that's fine. All we care about is what are these necessary conditions. And the sufficient conditions for what counts as life, that could keep changing as life evolves. But for the purpose of this talk, what I'm going to say is this, you have a concept of ecology and you have a concept of evolution, and you need both of these concepts to have a concept of life. So the necessary conditions for something to be called life is whether there's an ecology and whether there's some notion of evolution. And in the talk, I will of course make all of these very precise. So there's not just one kind of life. There's a social life, there's economic life, biochemical life, and so on. And you could ask similar questions about all of these. So for social life, you could ask questions about what is the ecology and evolution of languages, for example. You could ask many more questions, but you could definitely ask this question. For an economic life, you could ask questions about markets. The thing that we are interested in is particularly biochemical life. So we will ask about ecology and evolution of polymers. So the high-level summary of this thesis is I will show chemical reaction networks modeling an ecology of autocatalyzing polymers that are capable of open-ended evolution. And again, all of these terms will become precise. Yeah, so this is the thesis question. So can long polymers spontaneously arise and evolve? Okay. Now I will get into explaining something about chemical reaction networks themselves. For people who are joining later, there's fruits in the bag. Feel free to eat some. Okay, at least you will have gained nutrition by the end of the term. Okay, so what's the basic setup? What are chemical?
Sorry,
Yes,
provoking
please.
question later. So
Yeah, yeah.
probably know the answer to this question. Maybe it should be phrased like how long polymers spontaneously rise and evolve. I mean, we are here, so we know that they can evolve.
Yeah.
when they rise,
But
maybe the question is how.
yeah, but I mean, if you directly jump to the question of how, you would be excluding the possibility of alien life, that life came from some other planet, and you would be excluding the possibility of God. So I'm not saying that either of them are true.
I'm happy to exclude the latter, just funding supporters.
but so
But no, even
yeah,
for the first one,
Mhm.
okay. I mean, the same question would apply if the life came from
Somewhere
outside.
else,
So
yeah.
it's still then it follows, it can emerge.
Yeah. But I mean, like the thing is, and this is jumping forward to what is the thing that I have done, is I can at least show mathematically there can exist these chemical reaction networks where you have this thing being true, that long polymers can arise and evolve. But now, if we ask a question about how, then we would have to actually go into the geochemistry and biochemistry and actually say, this is the physical mechanism by which it arises. And I don't want to get into that because, because we want to like sort of just ask this question probabilistically, whether it is even possible, do we have a formalism in which we have these long polymers arise and evolve? So that's the scope, yeah. But thanks for the question. Okay, so what is going to be the setup for the talk? Chemical reaction networks, right? So the idea is, suppose you want to make you have this bottom-up approach to modeling some sort of processes, and in our case, biochemical processes. What are the things that you need to define? One, we want to define the types of all possibly infinite irreducible entities or species. Okay, so species is the technical term. And notice these are pragmatically irreducible. as opposed to irreducible in the strict mathematical sense. So what is an example of that? Suppose you want to talk about, rabbits and wolves in an ecology, then your pragmatically irreducible entities will be rabbits or wolves or cells and proteins. You don't have to necessarily go down to the subatomic level, although if you're looking at those high enough energies, then that's what you have to do. Then those will be your list of species. The second thing that we need is, so once we have the list of species, and the idea is that any individual of a particular type, they're indistinguishable. All you have access to is how many there are of that particular type. So the other thing that we need is how are these species, how are the composition of each species changing? Okay, and these are called stoichiometric interactions. And I will show examples of this. But essentially, the only type of interaction that can happen is maybe a species of type A will react with a species of type B to give you something of type C, and so on and so forth. Okay, so
Praful,
yeah.
in these things, do you consider having something like enzymes which will help certain things happen? Only if you have proximity, do you consider interactions of that type, which are?
Yeah, so there's two things. One of them is the level. So this is just at the topological level of what are the rules. But then once we get to kinetics, then we will make very particular assumptions about how can something like kinetically go forward. Like this is just saying
So
these are the
you're
possible
recording
rules.
all of these in some kind of transfer functions go from one species to another.
Yeah, right.
Okay.
So this is typically maybe connecting to Shahara's question. So these are typical like reaction diffusion models. They react, diffuse like this, or so physical propagation.
Yeah, so in stochastic chemical kinetics, the exact model is this, is that you say that here are the species and you need, through random encounter, some of them will come together and they will react to form, they will transmute to a different set of species. And the assumption that we will make will be that it's in a continuously stirred tank reactor. So, so your density is homogeneous everywhere. So there's no heterogeneity in that. Now, if you do want to make a reaction diffusion model where you have different sites with different concentrations, then you can also do that. But
sites we can consider continuous models.
yeah, I mean, yeah, and then you can take lattice spacing to 0 or something. Like you can make a lattice and then take the lattice spacing to 0, and then that would be the continuous version of that. So we can also do that, and that is also like the thing that you would.
Are these like conceptually different models?
No, because the only thing that will change is that you will need to index the species by their spatial location. So it's almost like having a field theory, but conceptually it's still a CRN. But it's just the CRN with this extra continuous dimension in that case, and then yeah. Okay, so so mathematically, you can also define a CRN, in terms of the set of species, complexes, and reactions. So let me show you an example of how this works out, and then and then it'll be very clear what that is. What this. So suppose you're looking at nuclear fusion in the sun, right? So this is the CRN. This is the chemical. I will be abbreviating chemical reaction network as CRN in the future. So this is the CRN for nuclear fusion in the sun. Now let's just take the first process, for example. So you have 2 protons coming in to make a deuterium, and then some neutrino, and then a positron. So this is an example of a reaction. And here you can see that a multi-set or a collection of species come together, a collection of species go out, right? So this is how you specify a reaction network. A reaction network will just be a collection of all of these reactions, okay? And the input, the source and target of the arrows of the reactions, those are called complexes. So so to define a CRN, you can define the set of species, the set of complexes, and all the reactions that happen in between them.
So in that model, the overlap probability of those 2 protons is entirely dictated by the temperature and density, right? In what you're doing also, you have some additional conditions that you have to assume for overlap probabilities, right? For these things to.
Yeah. So yeah. So this is good. so the assumption that we will make, so suppose we wanted to make like some sort of kinetic version of this, what we would say is, let's say we have a box which has 100 protons, and let's just focus our attention on the first reaction. Then we want to ask, what is the possibility that two protons come together, right? So it's like picking one thing out of, and there could be more things too, but it's essentially you will assign a probability to each of the reaction happening, and then the reaction will happen with that probability. And the way you assign the probability will be by making some sort of physical assumption, like if you're picking one at random, you have this much chance of doing it, like 100, and then there'll be some rate constant associated with it. So the thing is, like, I don't yet know, and I think it's actually a very big question of how to derive these from quantum mechanics itself. Like, I know Eric, for example, has papers on this, and other people have papers on this. So it's not as trivial as one might think. And those are like not details that I've exactly looked into.
But at the quantum mechanical level, just at the better level, we just calculate probabilities of scattering, right? So
Yeah.
why this wouldn't work even on the classical level? Let's say we take some Boltzmann type description. So that would be the way to calculate these probabilities, no?
Yeah. I think that is the way you would get to this through quantum mechanics. It's just that you would have to make those models, and I think that's where the trickiness comes in. That you would have to say, like, decoherence is happening, something, and you'd have to make all of those models. Right. But mathematically, CRNs are hypergraphs. So what is a hypergraph? A graph is you have vertices and then you have edges between vertices, and these can be directed edges. A hypergraph is you have vertices. So let's say you have vertices A and B, but now you have edges or hyperedges between collections of vertices. So for example, this represents the collection 2A plus B, right? This represents the collection A plus B. So for example, this edge is A plus B goes to 2B, right? So this way you can see that a CRN is, you can represent it as a hypergraph, and that's another way to see this. What a CRN is. And I will use this terminology. Well, I've already said CRNs can be found, in nature. You can think of just particle scattering. You can represent it as a reaction. Of course, there's an example in astrophysics, and there are examples in biology and chemistry, and that's why they're chemical reaction networks. Okay, so now what is the thing that we can do with the CRN is, as I said, these vertices or these species, they will have some population. So we call them species composition. Okay, so let's collect how. So suppose you have species from 1 through S. So you have N1 of the first one, N2 of the second one, and so on and so forth. And you can make a vector, and this is going to be your species composition vector. Now, given a CRN, you can say that suppose you want to make this reaction happen kappa 1 number of times. This reaction happen other number of times. What you can do is you can plug in the flows on the edges and then use this thing called the stoichiometric matrix to get change in composition. Essentially, you will see how these composition changes due to flows. So here's a more sort of worked out example of that. In the CRN that we had earlier, you say we have these 3 reactions. Now, if you say that this reaction needs to happen kappa 1 times, this reaction needs to happen kappa 2 times, what you can do is you can just collect everything. You collect the CRN in some sort of matrix, which is called the stoichiometric matrix, multiply it, or look at the image of the and by the way, the terminology for having a particular reaction go a particular number of times, I will call it like hyperedge flows. So for a given hyperedge flows, which is telling you how much you want each edge to have a flow on it, that'll give you a change in species composition, and essentially this is the sort of takeaway from this machinery that you will get. Oops, what did I do? Okay, so you will have a bunch of flows on your edges, and you will take the image of that under the stoichiometric matrix, and what you will get is how are your number of vertices changing, or how is your species composition changing?
Does this S depend on mean values of n?
Does this, sorry?
Does the S depend on the current mean values of n?
No, the S only depends on the hypergraph or the CRN that you specify. But So what can happen though
Why?
is that yeah, sorry.
Why?
So okay, so for example, if you say, let's take this reaction, which is A plus B goes to 2A, right? So if A plus B goes to 2A, if that happens kappa 1 times, then you're
But why doesn't depend on the number of A's and number of B's? It could be a non-linear function.
exactly. So all of that non-linearity, it comes in these hyper-edge flows.
So they do depend on.
Exactly, exactly. So the stoichiometric matrix is only a property of the hypergraph, but in general, these edge flows, they can depend on how many on the population itself, and that's where the nonlinearity comes in. Right.
So in this mechanism, you don't have ability to relate the, let's say, density of 1 versus 2 versus 3 kind of a thing in there, right? There are 2 species, I understand, but you don't have any dependence on one or the other. It's completely independent the way I see it there.
Yeah. However, these edge flows, as Professor Lukashenko just pointed out, these could have a very nonlinear dependence on the population. So this one could have dependence on 1 and 2 in general.
Okay.
So that's where the nonlinearity.
So it
comes
could
in.
be that this reaction will stop for whatever reason, and then you don't have evolution to whatever.
Yeah, that's also, yeah. And we will see in stochastic chemical kinetics, it is such that if your concentration goes to 0, the reaction will stop, like just by construction. Yeah. Okay, so that was chemical reaction networks. The takeaway is CIN is a hypergraph. It's stoichiometric matrix, transforms flows and hyperedges into those and vertices. Okay. Any questions? Okay. So now I will explain to you what we've defined as cluster chemical reaction networks. Now, just like a bit more about CRNs in general. So we saw that, a given hypergraph will tell you under a given edge flow, how does your species composition change? So the question then is, suppose there was some vector M that you could take the inner product with your change in species composition. And if M, the inner product of this vector M and the change in species composition was always 0, then that would mean that this M is, somewhat perpendicular to this change in species composition. And the terminology for it is M is called a conservation law, and the inner product of M and N is a conserved quantity. So if at some point in time, if M dot N, it takes some value, then it'll always have that value, no matter where you go. And if all entries of M are non-negative, then M is called a mass-like conservation law. That's because to each species, you're assigning a positive quantity, and that's like its mass. Now, contemplate this, that all real chemical reaction networks and that are happening due to actual atoms and molecules, unless there's some decay, radioactive processes or so on happening, you will always have a conservation of the number of atoms, right? The number of atoms of each type will always be conserved. So every real chemical reaction conserves the total atoms along a reaction. So this is the reason why we define this thing called cluster chemical reaction networks, where it has 2 properties. One, it needs to have at least 1 conservation law. And property 2 is all its conservation laws have only non-negative integer entries. Okay. So the idea is to each species, it's like assigning a chemical formula to each species. And some examples, so these are some parts of the Formose reaction. And you have all of these big molecules, but all you could do is just count the number of carbons that each of them have and represent the molecule with just the carbon. And that will be like its cluster representation. Now let's talk about the thing that we have to get at over here is about, we want to understand some form of polymerization, right? So for polymers in one monomer, you can think, okay, this is one monomer. Now 2 of them come together to get a dimer, 3 of them come together to get a trimer, but then you might have different geometries by which 2 of them come together. And then 4 of them could come together to make a former, but then you will have a different geometry. Similarly, for polymers in 2 monomers, of course, you can also have different geometries, but now if your monomers are A and B, you will also have different sequences which correspond to different polymers. All right. So yeah, that's it. So what we will do is, to sort of simplify things a bit, we will consider the cluster chemical reaction network of these things. And what that means is instead of, referring to a monomer or a dimer by each time, we will just represent it by the number of these conserved quantities that it has. So in this case, it's just how many monomers it has, right? So for example, this is a 3, this is a 3 prime, and so on and so forth. And this would be 4 because it has 4 of these. And if there are multiple things or different geometries, you can always distinguish them by putting a prime in their species or whatnot. But the simplification that this model allows you to do is now you can enumerate the set of all reactions as simply, there's some constraints on how integers add, right? So you will have all reactions, all second order reactions are of the type n plus m goes to L plus k, where n plus m is equal to L plus k. So what are some examples of this? So you can, and as I said, this is something that you can completely enumerate out in detail. So for example, 1 plus 1 to 2, and everyone knows this, right? 1 plus 2 is equal to 3, 1 plus 3 is equal to 4, and so on and so forth. So this then becomes a chemical reaction network, which we are calling the cluster chemical reaction network. And this is explained further in this paper. But and we will keep coming back to this. I'm just sort of introducing some concepts here. And then for monomers, for polymers and 2 monomers, what you can do is instead of having to remember the arrangement of the bonds, right? So whether it's BAA or AAB, those are different bond structures, but you could forget all of that and just worry about the conserved quantity, and you could map that onto a lattice. So essentially, BAA or ABB will both map to the cluster, which is 2, 1, which has 2 A's and 1 B. And in this way, this cluster chemical reaction network, it offers you a simplification that you're going from an exponential species space to a polynomial species space. So, just forget arrangement, just count. This is how you get to a cluster chemical representation of a CRN. Now, why are we doing this? Well, biochemical life is very complex, right? So if you're thinking about, DNA, you already have 4 monomers, AGTC. If you're thinking about, and then so many polymers, right, that come out of it. If you're thinking about vesicle fission or fusion, people make a very difficult. And this is how it happens. So it's as difficult as it needs to be. But what this cluster chemical reaction network representation allows you to do is it allows you to really simplify things to the bare minimum. So for example, if you have a notion of templating on your polymers, you can make a version of that with cluster chemical reaction networks, which will be somehow much simpler. Just because you've reduced the species space from exponential to polymer, that will give you some simplifications. And fission and fusion will just be given by these things. And in some sense, the beauty of all of this is it's a rule-generated chemical reaction network. So you can then say, give me all the reactions that happen up to a cluster size of 10 or something, and then you can just enumerate all of them. Oh,
so when you reduce it from D to the N to N to the whatever that
N to the D. Yeah.
So you lose some information, right?
Yeah.
So what's the kind of information that would be lost?
Yeah. So the thing that is the way we actually make this reduction is what we're doing is we're mapping things to like an equivalence class, essentially, right? So AAB and BAA, they're different things, but then they're getting mapped to the same thing. So the information that will be lost is if these 2 things actually behave differently in the original chemical reaction network. But the thing that this thing also allows you to do, yes, we have reduced all of this information, but we can always add information back in if it's necessary by adding new species at a time. So if you remember over here, I could always add back a 3' and then have more reactions with 3' if I need that information. And this way, I can first simplify it, but then gradually complexify it as and when I need it, right?
Can I try to translate all of this to some kind of physics language, and you will tell me whether I'm on the right track or not. So you started with this S matrix and species and A and B as an example. I would say this is like a kinetic equation. So things scatter and change. And now you're trying to reduce it to a number of conserved quantities, which would be like a aerodynamic description. You're not interested about individual microscopic scatterings, but you look at the more macroscopic conserved quantities. Am I sort of correct on the general physics language behind it, or?
Yeah, from how much physics I know, that makes sense. But yes, yeah. And those are those are the intuitions, I think, why we also want to, do this define these things. Okay, so we will return to this at a later point, but with the discussion so far, we will make a very explicit model for polymerization in one monomer, right? And it will take the form of this cluster chemical reaction network. Okay, so remember the title is mathematics of evolving ecologies and so on and so forth. So now we've spoken a bit about the math. We spoke about what CCRNs are. Any CRN with an atomic description, where atomic is not to be taken in a physical sense, but a more mathematical sense. And we can model polymers using CCRN. Now I will explain the concept of ecology. Okay, one of the key sort of these defining features, these necessary requirements on what life is, was put forth. And I'm sure there are more people, but from what I know, this is attributed to Louis Pasteur, that life comes only from life, right? And this is phenomenologically true. Like, you can verify this. Look around, yeah. So, but then you will be stuck thinking about this if you think too hard. What are we doing when we're talking about the origins of life? Because if life needs life to come, then what is this? And then you run into this chicken egg problem and so on. And the thing, and to me, like, this thing is now clear, is the key point to understand is not to worry about what came first, what came second, but the key point or the defining feature is that life makes more life, right? So in the sense that it's not about what comes from what, but the idea is that when you put both of them together, they'll make more life. So for example, in the case of chicken and egg, hen and egg, or you could just think of a child and an adult, you can make some sort of chemical reaction network, right? So you can say an adult gives you an adult plus a child becomes an adult, right? So then you can assign them some hyper-edge flows. You can make this one happen twice, which means a hen gives 2 eggs and one of the eggs become a hen. And in the process, now you have more eggs and you have more hen, right? Now, in the case of chicken and egg, and in the case of life as we know it, sort of this thing happens vertically. You have a parent which gives an offspring, but it's not necessarily that was always the case in an origin of life scenario. But what is important is the fact that life makes more life, and this is captured in the concept of autocatalysis, where autocatalysis is the process by which a combination of reactions involves a set of species which all increase in number, conditional on species in the set itself, while other species undergo a turnover. Okay. So in the high level picture where I'm going at is a CRN is an ecology if it contains a collection of autocatalytic subnetworks. Now, of course, I haven't told you what autocatalytic subnetworks are, so we will do that. But and we will come back to this discussion at the end. So what is the mathematics of autocatalysis? Suppose you have a CRN, a chemical reaction network. A subnetwork G will be called autocatalytic in the species set M. So M is the autocatalytic species. If what? If 2 things are satisfied. So first, let's look at the intuitive explanation of what we want, and then we look at the mathematical description. So the intuitive explanation, point number 1, is for some combination of reactions, at least each M and each element in M is consumed and strictly more are produced. Okay, so there should be some reactions through which each autocatalyst is used up, but strictly more are produced, right? And that's why it's autocatalysts. The second thing that we demand is that reactions cannot occur if none of M are present. So if they're just being produced spontaneously, then that sort of defeats the purpose. They're not autocatalyzing. So the mathematical description of these things. So for some, so there exists a flow on G, and that's the kappa, such that you have, the composite reaction takes this form, F plus some stoichiometry on the set M goes to stoichiometry N on set M, where 0 precedes M, which precedes N, right? And then the second thing, the second constraint, which is reactions cannot occur if none of M are present, is that for every reaction, the input complex Y contains at least 1 species from the set M, right? And that is because if none of them are present, then it wouldn't go forward.
I'm a little bit confused here.
Yeah.
If you start with a very simple CRN, let's say, if it doesn't have M in the species at all,
Yeah.
it's still possible that there will be a new chain of some kind that you start with. And that could be something that that could be autocatalytic, right? Some chain that I don't know about.
Yeah.
It's not there initially, but you might have created in the process of this reaction chains.
Yeah.
And then it becomes interesting enough that you start with nothing, but it ended up there.
Yeah.
Is that what you need in order for something to come out of?
Right. I mean, yeah. So like two points I want to make here. If you're actually modeling real life things, then it will never be that a bacteria or something just comes about into being without the presence of another bacteria, right? Like you need other.
Just like you were, something very simple.
Exactly, and that is exactly where I will be going. Is that for polymers, this does not have to be the case, because for polymers, there's two ways by which they can be forming. They could just be forming by getting bigger, but they could also participate in autocatalytic subnetworks, and then those dynamics become like super linear and actually interesting from the point of view of origin of life. And that is yeah. Like,
but you have to emerge to that. I'm saying from something simple also, you could possibly emerge to that.
yeah. I mean, monomers can make polymers simply. Like you don't need a lot to make polymers, I think, even in lab. Yeah. So those can spontaneously arise. That is true. Yeah. Yes.
I was going to just point out that maybe the example used of the chicken and egg isn't great, because chickens don't need to die to make eggs.
Yeah.
So one is not strictly true. Really what an adult is, because that's the same adult, we would say.
Yeah. Before
and after. So we would say that the adult catalyzes the conversion of food into eggs.
Yeah.
It doesn't actually convert into a new adult and a new.
But I mean, adult to adult just means like it's like a catalyst. So
yeah, I'm saying you have to frame it as a catalyst, because it's not consumed is the problem. Your intuitive statement, your first one is it's consumed, but it is not consumed.
I see what you're saying.
He just wants you to put food in there.
Well, no, just I mean,
the
example you gave it a catalyst.
No, but I mean, but the fact that it is consumed, but it's also produced, I would say. Like in net, it is not consumed, neither produced, but the fact that it occurs on the source side of reaction network, that's what counts. So this we would call as consuming a child, this consuming adult, but it just creates the adult bag.
Well, I agree. You know,
my biologist friends are laughing.
Yeah, if an adult has a child, the adult changes. So
yes, adult crime. Okay, so right. So now I have defined a few things. I've defined what autocatalysis is. Now there's a more restrictive notion of autocatalysis, which is called stoichiometric autocatalysis. But the beauty of this is, like, and actually it's, as David pointed out, the definitions are sort of clunky and there's things to worry about. But if you define a more restrictive notion of autocatalysis called stoichiometric autocatalysis, where you have this autonomous condition that each, it's that each reaction has a distinct input and a distinct output from the autocatalytic side. What this will allow you to do is now you can completely define autocatalysis by the stoichiometric matrix, right? So you have now just conditions on the stoichiometric matrix, and now you will see that we can use this to do things and then prove things and so on. And just to point this out, for second order chemical cluster chemical reaction networks, which actually have a conserved quantity, stoichiometric autocatalysis is the same as autocatalysis, but this is not this is not too much of an important point over here. Let's quickly just look at this example of autocatalysis. For example, you have, you can either look at that description or this description, but this is much easier. So 1 + 2 goes to 3, 1 + 3 goes to 4, 4 goes to 2 + 2. So now you can see that if 1 is provided as food from the input, then 2 makes a 3, 3 makes a 4, but 4 makes 2 2s, and then 2 will then make more 3s and more 4s and so on and so forth. So 1 is taken as input, but this whole thing, but the rest 2, 3, and 4, these are autocatalytic species. And together, this network becomes an autocatalytic subnetwork. Okay, in the autocatalytic set 2, 3, and 4. So what is the thing that we can do with autocatalysis? by the way, or stoichiometric autocatalysis, by the way we've defined it. So we can take a subnetwork, we can look at a stoichiometric matrix, and then what we can do is we can just categorize or partition the species into these subsets. So you have now a food set, a waste set, and then you have an autocatalytic set, which is then further, differentiated between a core part, which is necessarily increasing in number, and a non-core member, which may or may not be increasing in number. And the reason why you can do this is because you can just look at the stoichiometric matrix and you can see, okay, does each row have only negative terms, only positive terms, and so on and so forth. And this way you can, make this distinction. Now, what this allows you to do mathematically is, since we define autocatalysis by a condition on the stoichiometric matrix, so we say that, S kappa succeeds 0. What this does is it defines a cone on the edges, and then you can look at the image of that cone on the species composition, and that'll give you a different cone. And then you can define things about these cones, and you can prove things about them. I will not be going into them, but I just want to say that small autocatalytic networks or small chemical reaction networks can actually have a lot of autocatalytic subnetworks. And this is work that we've further done in this paper. And we can actually show that as you increase the size of this CCRN, we just say, instead of taking all the clusters up to size 4, you can take them up to size 5, up to size 7. And it's actually an exponential increase in the number of autocatalytic subnetworks that we have. So this is very interesting because the thing that we're relating with life, autocatalytic subnetworks, if we say that each autocatalytic subnetwork is like modeling a different type of life, then we can clearly see that we have this exponential, space in which, different networks can be, and then they can arrange themselves and so on. I'll talk more about this in the future. And there's some work that I've done with my collaborator, Victor Blanco. Now back to what we're trying to do over here is define a concept of ecology. So the collection of autocatalytic subnetworks, I will define that forms an ecology. And actually, there's more reason to say it than just what has come before. Like, so far, I've given you this conceptual reason why that works. But also in a previous paper, what we've shown is that you can now compose different autocatalytic cycles in different ways and get different ecological behavior. So for example, if two autocatalytic cycles are competing for food, then that will give you some kind of ecological behavior. And a similar thing is shown over there, although this is more fun. If the food of one autocatalytic cycle is a different autocatalytic cycle, then you have some sort of predator-prey dynamics. You can get into limit cycles and so on. So this is like, I mean, the idea over here is that you can, this is just a conjecture at this point, I guess, you can get all sorts of ecological dynamics by just composing autocatalytic networks, right? And so that's the reason why I'm equating the concept of ecology to just a collection of autocatalytic subnetworks. Now, back to the question at hand, we want to talk about polymerization in one monomer, and suppose in this the pre-life Earth, you have some monomers which are floating around. Now these monomers can form longer polymers, so 1 + 1 can be 2, 1 + 2 can give you 3. You have this whole chain up to n. But now suppose you have an external, just a production, a source of these monomers coming in from your environment, right? So they can come in, they can go out freely, there's some reservoir. And we say that suppose there was now one of these polymers, they could actually act as a catalyst for monomer production, right? And this actually has a biological significance. There's this thing called kinases, and they serve the same purpose in their ecology. But for now, let's just say that there's a catalyst, C1, and what it does is it catalyzes the production of more monomers, right? So, but suppose you do that, then, I mean, I can just, by the definitions of autocatalysis that I've shown you, I can show you that these two reactions, so C1 - 1 + 1 to C1, and C1 to C1 + 1, this forms an autocatalytic subnetwork. And you can, just compute this, like 1 goes to C1, but C1 makes C1 + 1. And of course, no one will make more C1. And I've only sort of cut the chain off at C1 - 1, but you can actually see that if you extend this back to all of the polymerization reactions, then the whole set up to C1 will be an autocatalytic set. So what this catalyst does for us is, it adds a minimal autocatalytic subnetwork to the CRN, and hence it makes it an ecology. And this is a sort of, the intuition to have over here is that this polymer now has a functional role in the ecology that it did not have earlier, without that particular reaction. You can do the same thing for two monomers, but for interest of time, let's say that you agree with me. Well, the terminologies is that my polymers in two monomers, they're labeled by I, J, and then you can have similar catalysts and so on. Okay, so that's the concept of ecology. A collection of autocatalytic subnetworks is an ecology, and polymer CCRN interactions naturally form an ecology. And by naturally, I mean in two ways. One is because, as I've shown you, right here, that these are very natural to polymers, right? This is how polymers can add and break up. So of course, there's a lot of autocatalytic networks over there, but also you can have these extra catalysts in the space of polymers, and then that further makes it into an ecology. Okay, so I hope no questions. Okay, so I'll move on to stochastic chemical kinetics, and I promise you this is the last mathematics skill part, and then we will quickly, we're coming to an end. So in terms of stochastic chemical kinetics, the whole sort of pre-defense talk was dedicated to showing how you can start with probability distribution, and then you have this notion of Markov process, and you can represent it, and you can massage the equation into something that looks like, something like your Schrödinger's equation without the I, right? So d rho, where rho is now probability density, and h is your stochastic Hamiltonian. But just to sort of, bring things back, the intuitive understanding of stochastic chemical kinetics that I would like you to have for right now is, as we said earlier, there are a lot of reactions that can happen, and that is given by a chemical reaction network. Now you take some sort of you make some sort of random encounter model where things are just bumping into each other, and when they bump into each other, with some probability, some reaction might happen, right? So you will get, if this is your population space, your counts can go up and down, and stochastically. Now, some things that can happen is that they can have attractors, and we will get to that. But one thing that will generically happen for these things that are given by continuous time Markov process is that eventually, if you just let them evolve for a while, and you can look at this stochastic simulation, then you can look at its histogram of how much time it has spent at each point, and you will see that this, as the simulation runs for longer and longer, this converges to what is called as the stationary probability distribution, okay, which I'm denoting with pi. And since it's stationary, the rate of change of the stationary distribution is 0. So what this means is, just plugging it back into the equation from the last page, so the stationary distribution is the 0 eigenmode of your Hamiltonian operator. Okay, that's how it's defined. Now, I also showed you in the last time that we can get analytic sort of results for this, for what the stationary distribution looks like in some simple cases, and the sort of connecting factor over here is the equation down below, which is that you can represent the probability distribution asymptotically as exponential of minus v times math cal v, which is called the non-equilibrium potential, and we can and we also have an algorithm to compute what this math cal v is, but mathematically this non-equilibrium potential, okay, this math cal v, which I will now call as the non-equilibrium potential, it is nothing but a solution to your Hamilton-Jacobi equation on the h equal to 0 submanifold, right? So this is something that we discussed last time, and so the idea is that now let me tell you some facts about this h equal to 0 submanifold of the Hamilton-Jacobi equation, right? So firstly, for chemical kinetics, for the model, for this random encounter model, you can write down a Hamiltonian for your CRN, and it takes this form where you have a sum over all of the reactions, So for each interaction, you will have a new term. And there's an exponential dependence of the momentum. And there's some polynomial dependence on the concentration or your composition variable, normalized with some factor. Now, first, like right off the bat, you can see that if you are looking into the H equal to 0 submanifold, if you plug in P equal to 0 over here, this satisfies H is equal to 0, right? And then you can find the equations of motion at P equal to 0, and you will get dq by dt is del H by del P at P equal to 0. Then you just plug in this Hamiltonian, and you will get something of this form. And to people who recognize this, actually everyone in high school, I'm sure you've seen a version of this. This is the mass action kinetics. So this is how you can derive the mass action kinetics from H equal to 0 Hamilton-Jacobi solution. But the other interesting thing about this, about these solutions, is that so the non-equilibrium potential is defined precisely by the solution where momentum is not equal to 0. Okay, so your partial derivative in Q is not equal to 0. That there will also be some solution in this H equal to 0 submanifold where momentum is not equal to 0. And if you integrate out that momentum, then you get the non-equilibrium potential. And in fact, there's a very cool thing that you can prove, and this is shown in this paper, although people have done this for ages. But you can show that the non-equilibrium potential is actually a Lyapunov function with in the mass action kinetics, right? So the gradient of nu dotted with dq by dt at p equal to 0, this is always less than equal to 0. So what this does for us, or what this intuitively means, is something I will now explain. So first, let's try to get a better understanding of how to look at these chemical reaction networks and their dynamics to begin with. So suppose you have, just let's take a one variable model, and you have mass action kinetics, and you have a velocity flow field given by the mass action kinetics, which are plotted here in blue. Okay. Now, if you start over here, your velocity is greater than 0. Okay, so you want to go more to the right, right? So you go towards the red point. If you start to the right of the red point, then you go back, right? Because your velocity is less than 0 and it pushes you back. So, and if you are at the red point, then that's the velocity is 0. That means that's a fixed point. And by the other considerations that I just mentioned, it is a stable fixed point. So a stable fixed point is what we will call as an attractor, because you can move around it, but you will still come back to that point. Now, correspondingly, you can look at the gray point over here or over there, and you can convince yourself that if you're to the right of it, you want to move away from it. If you're to the left of it, you want to move away from it. You move away from it in every direction. That's why it's called a repeller. And so this is the idea that your mass action kinetics will give you some polynomial, which you can then plot out, and you can figure out what are its fixed points. You can then do more analysis to figure out what are the stable fixed points, where are the attractors, where are the repellers, and so on. And another thing that you can now do, because you have the Hamiltonian, is you can find out what the non-equilibrium potential is, right? Because it's the other solution in the H equal to 0 submanifold that we did not consider. Now, given the fact that this non-equilibrium potential is Lyapunov with respect to mass action kinetics, what does that mean? What that means is, for mass action kinetics, of course, if you move from the right of the repeller, you go towards the attractor, right? So that means the non-equilibrium potential must be reducing, right? It has to reduce when you're going with mass action kinetics. So what that means is at the repeller, your non-equilibrium potential is higher than what it is at the attractor. And this is true for any repeller and attractor which are neighboring, right? Which is just guaranteed by the mass action, by the Lyapunov property of this non-equilibrium potential. What, however, we don't understand is, or we don't know a priori, is what is the relationship of non-equilibrium potential at one attractor to another attractor. That's something we're not guaranteed. But if it's just a repeller right next to an attractor, then we're guaranteed that the repeller must be higher than the attractor. And I will explain more of why this is relevant in a bit, but I just want to say this for now, that what I showed you was a 1D case, but in fact we can do this for any dimensions, and we gave a functional gradient descent algorithm in that paper to do this for sort of higher dimensions. And okay, and that's what the pre-defense talk. In that we discussed that. So back to the polymerization in one monomer. Up until now, we only had looked at the hypergraph or the chemical reaction network. Now what we will do is we will look at the actual kinetics. So let's say each of these reactions happen with some rate constant, put in some rate constant. You will find some fixed points of this thing. You don't need to look at it. For some choice of rate constants, I've plotted what the fixed points are. Okay. So if you're for species 1, 2, 3, 4, remember this corresponds to polymers of size 1, 2, 3, 4. Now you have one attractor where you're and this is plotted in the log scale. Notice. So you in one attractor, your population of size 4 polymers is very low, is exponentially lower than what it is in the other attractor, right? And then you have this repeller in between as speed. So now for this choice of model for which I've shown these attractors, I can then use the algorithm, the FGD algorithm, to find what the non-equilibrium potential looks like. And what I find is that, of course, this is by construction, but what I find is that, so suppose I plot this against the mean polymer length. So notice the mean polymer length of this thing is lesser than the mean polymer length of that one. And so this is the lower attractor, and this is the higher attractor. And for this choice of kinetics, I've shown, or you can sort of verify this, that the non-equilibrium potential is higher at the lower, where your mean polymer length is low, and it is lower when your mean polymer length is high. Now, what all of this means is, remember back to the equation where we had an interpretation of what this non-equilibrium potential is. The stationary distribution is exponential of minus the non-equilibrium potential. So what this means is the higher your non-equilibrium potential is, the less likely you are to be in that state in the stationary distribution, right? And you can see, put nu to infinity, you will see this is 0. So and then so what I've essentially shown is that this state, which has a bigger mean polymer length, is more probable than the attractor with a smaller mean polymer length, at least for the model that I've shown. And we will I will now situate all of this conceptually in the bigger narrative of origin of. But for now, are there any questions on this sort of conceptual or technical points? Okay, I see some nods, so I'm happy. All right, so now we will talk about sort of the last concept that I will introduce is evolvability. Remember, for life, we need two things, ecology and evolvability. So a bunch of new definitions, but I promise it'll be worth it. So evolvedness, what we do is we want to say, and this is, phenomenologically, the person who's making the model will have to define a vector valued function on the species composition space, which denotes the evolvedness of that composition. So for example, intuitively what we're thinking is this mean polymer length being higher is more evolved than mean polymer length being lower. But this is sort of where phenomenology comes in. But what you can do is you can say to each species, let's assign a species trait, and we can look at the average species trait of the composition. And this way we have some notion of what is the evolvedness of that composition, right? Where Q is representing the composition. Now, for the cases that we are doing with the cluster model in 1D and 2D, it's very easy to do these because remember, your species were already indexed by integers. So what you can do is you can find out what is the average cluster size in one dimension, or you can find out what's the average cluster size in each of the two dimensions, and you can define a vector valued evolvedness. Where the inherent sort of the bias that we're bringing in over here is we're saying the bigger the average cluster size, the more evolved that attractor or that composition is, right? Which is the place where we want to be for our purpose. Okay, so finally, what is our notion of evolvability? And since the term non-equilibrium potential is already taken and it has this mathematical sort of package, what we will do is we will define the cost of an attractor as the relative value of the non-equilibrium potential at that point. So, and this is, more intuitive, like if you are less costly, then it is more probable. If you're more costly, then you're less probable, right? Very intuitive. So for example, on a plot against evolvedness and cost, if you look at this attractor, which is less evolved but more costly, and over here you have something which is more evolved and less costly or lesser costly, and this is even more lesser costly, then you can see that if you started out a system really close to zero, it would find its way to the first attractor, but then it would evolve to the second one. And this is much more improbable than this way. So it'll go forward and then it'll sort of stay over there. Or, I mean, it's a stochastic system, it'll do whatever it wants. But the way we can sort of define this is, given an attractor, if there exists a more evolved attractor that is less costly than the current one, then the attractor will be said to be capable of directional evolution. So at least from an attractor where you can find another attractor which is more evolved and less costly, if that exists, then at least from that attractor, you can directionally evolve to this new attractor. And then, and if you can keep doing this infinitely, then that would be open-ended evolution. But I'm jumping ahead of myself. Yes.
I would have thought that to have a very evolved state, maybe I'm thinking wrongly here, but a more evolved state would be more costly to get to. That's why I would think that a very complicated evolved state would be less probable to have it randomly.
Yeah. So I mean, I think this is the contribution that I do is I disentangle evolvedness from cost. And the thing is, evolvedness is, it's also phenomenological, and there are all of these other processes. And cost is defined in purely a probabilistic setting, right? And that is the beauty. So if you want to ask whether or not life is inevitable, the kind of thing that we want to come up with, if we say, what we want to say is that this configuration in which we are right now, it is the more probable one, right? It's only then that life becomes inevitable. Unless, so what we actually want to get to is we want to say that there can be more evolved attractors which are very less costly, and that is true evolution. And nature finds them, and and that's that. Like, unless we can show that, we cannot say that life was inevitable. Do you agree?
Perhaps. I think more. To me, it sounds unintuitive in the sense that it should be more difficult to get to a very complicated.
Yeah, but then how is
Why
life
would that
inevitable?
happen? I understand,
Yeah.
but if I don't have an experiment which I can run multiple places just to figure out, I don't know if the life is more probable or less probable.
Yeah, but at least mathematically you can see that we have one example where the thing which is more evolved, right, longer polymers, they're also more probable, so they are less costly, and that is an example that I've constructed, right?
Just the existence is probably more probable, but the concentrations are probably not the same. That's probably what I was missing.
Yeah.
Right. There's a whole lot more carbon dioxide than the complicated molecules, right?
Yes. Yeah.
Well, yeah.
Okay.
It's probably true that biochemically, if we actually understand them, they don't turn out to be true.
I mean, you're defining it in a non-spatial, you're imagining one well-mixed system, but of course, the way the planet does it, you can have local evolvedness, maybe at the cost of global less evolvedness.
So, yeah.
Mhm.
Okay, so a few more things. Yes, our notion might be, my notion might be less evolved than where it should be. But with the way that I've defined it, evolvability might be generic in CRNs. And, and this is some ongoing work that I'm doing with my collaborator, Vlad. But the thing that we want to show is that if you could ask a question, given any set of attractors and the relative sizes on their, and the relative costs, suppose I specify these are the attractors, these should be the costs. Can you find such a CRN? And we suspect, like in 1D, we can do that. And we suspect in higher dimensions, we can do that. Okay, but anyways, back to the question. One monomer polymerization CRN is directionally evolvable, right? So by the way that I've defined directionally evolvable, I think you all will agree now that yes, if you were at this attractor, you will go to this higher attractor, and you can keep adding more and more catalysts and you can sort of keep doing this, ratcheting your way up. Another concept that's important for biologists is historical contingency. And the idea is that there are at least two more evolved and less costly attractors between which the system can only transition through less evolved and costly attractors, and the stochastic CRN will be set to exhibit historical contingency. What this means is, suppose you have an attractor set up like this. So you can go from this attractor to this attractor, and might be less, and suppose this attractor is less costlier than this one, and this one is also less costlier than that one. So both of the evolved attractors are actually less costly, but there's no way to just go from one of the evolved attractors to the other one, except through going backwards and then to that way. So this is an example of historical contingency, because you can now imagine having a tree of such attractors, right? Where you can go from one of them to the other one, but then you cannot go from this one to that one, and you can keep doing this. And this is the biological or biologist notion of life as we see it, as historically contingent on all of these choices that came before it, right? So for example, if you're over here, then we needed to have come from here to there, but then we needed to have come from here to there, and so on and so forth. So that's the notion of historical contingency that we can also formalize with our definitions. Finally, open-ended evolution. If at each attractor, there's one more attractor that is less costly and more evolved, right? Then this thing is open-ended. It's capable of open-ended evolution, because at each attractor, you can just find another attractor which is less costly and more evolved. You'll just keep evolving. And in our system, we can just keep adding new catalysts, and that will make it such that you can add infinite catalysts, and the system will become open-ended. All right, so this is we're getting to the end of this. Finally, I want to do a short discussion on the origins of biochemical life, just to tie things up. Here's a summary. As we said, this concept of life, it's sufficient, but not, it's necessary to define concept of ecology and evolution. And the concept of ecology is defined as collection of autocatalytic subnetworks. Then given an ecology, you can find the attractors. These attractors, by the way, are non-equilibrium steady states. So although things aren't moving, like the species composition might not be moving, but that doesn't mean that nothing is happening. It's like, you can think of yourself standing in a railway station or something, and the number of people could be the same, but people would be going in and going out. So that is the picture that you should have of an attractor. And then you can assign evolvedness to these attractors. Then using Hamilton-Jacobi theory and the algorithm, we can find the cost of each attractor, and then we can ascertain evolvability using the costs, right? So this is sort of the formalism that I've sort of given in my thesis for talking about these things. And then from the point of origin of life, mathematically, we show the existence of CRN models where a higher abundance of longer polymers is more probable, right? And other processes can now act on the long polymer attractors. So I should speak a bit more about this. Now, the hope over here, back to my thesis question is, yes, I was just asking whether or not we can have a formalism where you can have these long polymers, which are also more probable. But the hope afterwards is that once you have a sustaining population of these long polymers, then other processes can start acting on them, and then they can complexify to give life as we know it, or at least like make a transition from non-life to life. Finally, like just to summarize like the mathematical things, if you want to verify ecology, you solve certain linear programming problems, and we have an algorithm for this, and it's one of our papers. And then if you want to verify evolution, you solve some minimax problems, which amounts to solving Hamilton-Jacobi equations, and then we also have an algorithm of that. So yeah. So there are multidisciplinary achievements of my thesis. I clarified various notions of autocatalysis, contributed to like mathematical formalism, algorithm, and visualization for enumerating stoichiometric minimal autocatalytic subnetworks. And then I formalized the role of autocatalytic subnetworks in ecology and provided explicit examples. And then on the other side, I clarified the information theoretic and geometric connections of stochastic CRNs using Hamilton-Jacobi theory. So this is something that people actually care about. Gave a functional gradient descent algorithm for finding solutions to the Hamilton-Jacobi equation. I don't think people have done this in the past. So like to my knowledge, and then we formalize the role of all of these things in actually defining something for the origins of life and provided examples where that thing was true. So yeah, so you can see that it's like multidisciplinary. There's some applied math, some mathematical physics, some computer science. Yeah, functional gradient descent algorithms are, even I didn't know this, but they're like leading edge of computer science, what people do these days, which is really cool. And then of course there's contributions of origins of life. All that being said, because of questions that were raised earlier, like have we actually just defined these things, or have we come closer to talking about real life? Well, I will now speculate and sort of convince you that we have. So there's two pillars of modern biology, from what I understand. One is the central dogma, which acts on short time scales, and the idea is that information flows from DNA to RNA to proteins, right? So those are the actual biological machinery that's happening inside biological things. And then the other pillar of modern biology is Darwinian evolution, which acts on really, really long scales. And the idea is that you have some sort of mutation, inheritance, and differential success, and then the ones which are less fit get selected against, and so on. Now, through the formalism that I've given so far, how can we make some progress on this? And I will now comment on that. So first, let's look at the information flow from DNA to RNA to protein. You can also look at it as the space of possibilities, or the entropy of the bonds in a protein is controlled by the RNA from which it was translated. So information flow in one direction is like constraining entropy in the other direction. So the question that we could ask then is, what are the origins of chemical control systems? How do these control systems come into being, right? Because even if we do have a population of long polymers, what's next, right? How do we get to DNA and such like and so on? And then the idea is that some literature to be aware of, that strong Turing completeness of CRNs in the model that I've used has been shown. So you can actually configure them to do all sorts of computations. You can also have CRN implementations of neural networks. There's stuff done by people in our math department. So so it is very promising that actually CRNs might actually form the right set of primitives to be talking about these things and bring them to full generality. The question that we can ask from what has come before is, how does adding and removing autocatalytic subnetworks change the CRN dynamics, attractors, and the relative costs? And can we create circuit diagrams for CRNs using minimal autocatalytic subnetworks, right? Because it would be ideal to just say, look, this is the kind of control system we want. Go take some autocatalytic networks, make a circuit diagram, and then show that it has these properties, right? That would be really cool if that can happen. Secondly, as was pointed out multiple times, is that I'm only considering spatially homogeneous setup, and there's no notion of individuality without a spatial heterogeneity, right? Like you're different from me because so on and so forth. So there's multiple ways in which you could do this. You could put CRNs on a lattice and have these lattice sides diffused, where each lattice side is modeling one individual. You can also put them on a continuous lattice, and then you can say you can have these reaction diffusion systems, and you have these solitonic solutions. Here you can have cell-like patterns and Turing patterns and so on and so forth. This is all stuff that people can do. Well, the question that I am interested in with the work that I've just shown is, how does composing a CRN with other CRNs on a lattice, whether it be a finite lattice or an infinite lattice, influence the ecology and evolution, right? And once we can start answering that, I think we would get closer to having a notion of actual Darwinian evolution on longer timescales. So that's just a speculation. Yes, I want to thank Eric Smith, David Baum, my collaborators, Victor Blanco, Jen, Alex, Vlad, and the committee, and all my friends. Yeah, I really want to thank Eric and David right here. Okay, so thanks for this opportunity. There are more acknowledgements in the dissertation. Your name might be there. Well, now some shameless advertising. So I am going to be playing, or some of my friends and I are playing a show. It's Thesis Offense, as opposed to the Thesis Defense, which you are at right now. And it's May 20th, 21st. It's a weekend from 6 to 8 p.m. Also, we plan to clean James Madison Park, I think on the Sunday of 21st. So you should like bring your friends, bring your kids, bring whatever, whoever. Also, if you play chess, you should bring your own chessboard. We will also play a lot of chess. Yeah, and this is the library mall opposite Memorial Library. Okay, so yes, that's it. Thanks.
Typically, we do audience questions at this time. All right, so the committee stays and everybody else needs to leave. Same on Zoom, by the way. Eric stays.
Right, understood.
Can I ask a Zoom question or
Just, oh, sure.
is the time passed?
Yeah. Sorry,
John, yeah, John Yen.
yeah.
And there is a question on Zoom, if anybody can.
Thanks.
Ask-
Yes, John. Can you hear? Yeah.
Yeah.
Feel free to ask the question.
Yeah, basically, your theory, I understand, was developed for polymer compositions rather than sequences. And we know that life depends on polymer sequences, that they're important at least. So my question was to what extent you think your theory can be generalized to ecology and evolution of polymer sequences in the origins of life, rather than just polymer compositions.
Yeah, thank you for the question. So I think, this notion of evolvability that I've this does not depend on what is the system that we're looking at. All I'm saying is, if you have a CRN description, you can check whether there's an ecology. You can use our AFGD algorithm because all it requires is a CRN. So you could do this for any CRN, and you could figure out whether or not that CRN is evolvable in the sense that there exist other attractors that are more evolved and highly probable or more probable. But secondly, in the cluster chemical reaction network, the thing that I did, well, what I was doing is I was reducing the space from like exponential to polymer by not caring about the sequences. But I think the formalism itself is generic enough that if you wanted to put in the particular details of sequences, you could still do that and you could still go ahead with the analysis. Yeah, I don't think anything changes.
Very good, thank you.
Thank you so much. Yes, I see Eric, I think, has something to
Yeah,
say.
thanks, Praful. I want to follow with a kind of a mathematical, something like an articulation of John's question also, and related to Shitaru's point about what happens when you coarse grain. So I understand your proof that given a polynomial with a finite set of local minima, you can always design rate constants so that they're ordered, so that the one furthest out is the lowest. And you have in your dissertation as a conjecture that this can be extended indefinitely. But then I have a question, and this relates both to the tree of sequences as opposed to the lattice of formulae, and also a question that I think is of interest in biology. The non-equilibrium potential is bounded below at 0, because the most likely you can ever be is 1. And so I wonder if even if your conjecture proves true, that you can formally continue to have a sequence of minima that decrease, they must wind up decreasing by smaller and smaller amounts because they hit the lower bound. Otherwise, your starting point winds up having to become infinitely improbable as you extend your induction on these things. And whether that means that your one-dimensional model starts to look like what biologists call neutral evolution. Your evolution may be strongly directional in the early stages, but then become more and more neutral. But then the question is whether, if you add back the combinatorics of sequences, the reason you retain open-endedness is that you wander around in this combinatorics, and so your evolution takes on a very different character where undersampling and history dependence become the big thing. And I'm just curious sort of where you are on trying to extend the things that you showed for finite sequences of minima to your conjectured indefinite case, and then also whether you have ideas about how to bring some of these biological concepts in and maybe address them with the sorts of ideas you've already put up.
Mhm. Yeah, thanks for that comment. Just from what you've said, I can definitely analyze in this infinite dimensional case how the spacing of the non-equilibrium potential changes as the attractors go out to infinity. And from what you said, it does sound very logical that the spacing should be reducing. But I think that's something that I can probably like sit down for a week and verify. Yeah, but yeah, I mean, I hadn't thought about this point, but I think this is probably something that I can pursue in the future. In a sense, yeah. Thank you. Very good point.
Can I just follow up with that? So the implication then would be that while we might expect you to get higher and higher evolvement up until a point, at some point, you are in a much more kind of a flat landscape where which state you're in, which composition you're in, is going to be shaped by these it's like symmetry. It's like there isn't much pushing you probabilistically into one or the other composition, but that other factors like effect on fitness become more important. Is that?
Yeah, I think that's
saying?
yeah, I think that's the picture that's arising.
Okay.
Yeah, where like the sequences become actually important and so on and so forth. Yeah.
So I'm going to maybe connected physics.
Yeah. Well, by the way, should I send everyone else out except
except for the committee.
Yeah. So yeah, thank you everyone for joining. Now, now this is the committee only session. But if you have questions, please let me know.
Eric should stay.
Yeah, of course, Eric will stay. Eric, I'm hoping
Don't
you'll
close
stay.
the session.
Yes. Yes. Yeah, just 1 second. So let's see what people are saying. Okay. So I can also remove them. Okay, sorry, just 1 second. Maybe some people forgot that. So I'm just going to remove them real quick.